Aloha, everyone, and welcome to Biology 101 at Chaminade University. My name is Genevieve Griffiths, and I will be walking you through what we're going to be talking about for the rest of the semester. So this chapter is called An Introduction to Life on Earth, and we'll be discussing multiple different topics that we will touch on throughout the course of the semester. We'll talk about the definition of life, so what do we use as parameters to define what is alive. We'll talk about evolution, so what happens as organisms end up susceptible to natural selective pressures over time, so how populations are going to change under those selective pressures. We'll talk about how we study life, so what are the parameters that we use in science, what are our scientific method and scientific design, and what is science as a whole. And just to get started, we'll toss out some definitions. So biology comes from two Greek words, first one being bio, which means life, and logy, which is going to mean the study of life, when you put those two together. And there's multiple characteristics that we use to define life. And organisms defined as alive have these 6 major characteristics. One, they are going to acquire and use materials and energies from their surroundings to be able to drive their organized complexity. So they actively are going to maintain these levels of organized complexity. And what I mean by that are that cells are going to be organized into tissues, and then tissues are going to be organized into organs, and then organs into organ systems, and so they get more and more and more complex as we build the organism as a whole. So basically the whole is going to be more than the sum of the parts. Also, organisms that are alive are going to perceive and respond to external stimuli. They're going to grow, reproduce, and as a whole have the capacity to evolve. And I want to make a point here that it's not the individual organisms that evolve, it's going to be a population that is subjected to selective pressures that's going to be evolving. That's why they use the word collectively there. So the basic unit of life is going to be the cell, and the cell is what's going to keep the inside separate from the outside, right? So the cell is going to have an internal series of organs, which are called organelles, that are going to be separated from the surroundings by a membrane, and inside there we have the variety of structures. Again, those are organelles, and chemicals found in a fluid environment. What does that look like? So this is an overview of the cell, and we are going to get into more detail when we get to the cell organelle lecture, but for today we're just going to give you an overview. So inside of the cell we have what's called organelles. Just as your body has organs, the cell has organelles that are going to have specific functions within the cell. The largest of these is the nucleus here. The nucleus is, you consider this like the boss's office. This is where all of the information is going to be created for all of the work which is going to get done out here on basically the factory floor. And that's going to be everything outside of the nucleus is going to be considered the cytoplasm, which is going to comprise of the organelles, and the cytosol, which is this fluid in here. Outside of that, we also have a plasma membrane, and this is a plant cell, and you can tell because we have a cell wall, and that cell wall is going to be depicted here in blue. That's what's going to give it kind of its brick structure. Now, an animal cell does not have a cell wall. Another way you can tell this is the plant cell here, these organelles here, which are going to be chloroplasts, right? So the animal cells are not going to have chloroplasts. All right, we'll talk about all the different organelles that we can find inside a cell when we get to that chapter. But let's talk about what we use to define being alive. So being alive means one of the 6 characteristics is that you acquire and use the materials and energy around you to maintain what's called homeostasis. And you'll probably hear the word homeostasis a lot throughout the course of this semester, and homeostasis is basically the maintenance of internal conditions. And we need to use energy from our surroundings in order to maintain our internal conditions, in order to grow and to reproduce. And that means that we have to acquire important materials and building blocks from our external environment, things like minerals, water, etc., that are acquired from the external environment and brought into the organism. And they can also come from the other living organisms as they have started to decay. The organisms are going to need this energy constantly to be able to sustain themselves. This is just depicting one of the smallest organisms and just demonstrating that they too, even at the very lower levels of life, have these 6 characteristics. So here we have antennae and eyes, which are going to demonstrate that they're responding to the stimuli around them, and the gut, which is where they're going to be absorbing nutrients and bringing in food from their external environment and excreting waste, and then eggs, which are demonstrating that they also reproduce. And there's 2 mechanisms by way we obtain energy, but they all come either directly or indirectly from the sun. So organisms that undergo photosynthesis are going to get their energy from the sun. They're basically going to be capturing that sunlight and turning it into glucose, which is an energy carrier molecule that you're able to use when glucose is broken down, it releases a lot of energy that the cells are able to use to drive the metabolism within the cell. So some organisms are able to undergo photosynthesis and get their energy directly from the sun, and other organisms are going to get their energy from the consumption of the bodies of other organisms. And you've probably seen this in very basic science classes in 3rd and 5th grade. Here's what we call the food web. You can also call it trophic cycles or energy cycles. And what's happening is all of the energy is coming in from the sun, and that's going to drive primary producers, these plants, which are going to be creating energy during the process of photosynthesis. And then they are going to be consumed. Here's another set of plants being eaten by the zebra, and then that zebra is going to be eaten by the lion. So some organisms have to eat other organisms, and then some organisms eat plants. And all of these are going to be subjected to detritus, which is shown here in the purple cycle. So this is all organisms that are going to end up decaying, and eventually we're going to be losing energy into the environment, which means that we're going to have a constant source of energy coming in here from the sun. So living things are going to constantly be bringing in energy on a continual basis to sustain themselves. And again, we're using that word homeostasis here. There's a lot of different internal conditions that have to maintain an equilibrium. That means things like your body's pH or your temperature or your salt balance or any of the metabolites inside your body have a particular range of conditions in which they are supposed to be maintaining. And that means that if we have too much of something, we have to send it out. If we have too little of something, we have to bring it in. So we have these pumps on the outside of the cells, on the membranes, that are going to pull chemicals in or send chemicals out in order to have the appropriate conditions for chemical reactions to occur. And all of this as a whole is considered homeostasis. And if you fall out of homeostatic conditions, then you can end up with some sort of illness or disease state. All right, so organisms also, again, they're responding to the external environment. They can respond to things such as light, sound, chemicals, but they also have internal stimuli. For example, stretch receptors that tell you that you have to urinate because your bladder feels like it's full, or temperature sensors, pain sensors, chemical receptors. So you have a lot of internal stimuli that help you to maintain homeostatic conditions, as well as to alert you to things like injury or to just your overall body's status. But even lower level organisms like plants and bacteria also respond to stimuli. For example, plants are going to grow towards the light, and bacteria are going to move away from a toxin or towards available nutrients in a media. Another depiction of an organism being alive is that organisms grow. That means that all organisms are going to become larger over time. Consider a baby growing into an adult organism. And we do this by producing more cells in order to increase their mass. And that's through a process called mitosis. We'll have a whole chapter on mitosis and its sister, which is meiosis. Meiosis is basically the creation of haploid cells, or sperm and egg, that are going to then fuse together to create a genetically unique offspring. And that's a very specialized type of cell division. And bacteria are going to just grow by enlarging their cells until they get too large, and then they're just going to divide in half, bacterial fission. And that's going to happen after the genetic material has been copied, so they're going to be genetically identical to their parental cell line, kind of like a cloning. Either way, these growth is going to involve taking external materials that are acquired from external environment and converting them into molecules that the organisms need to be able to grow and survive. Additionally, to be considered alive, an organism must reproduce. And organisms can reproduce in a couple different ways. Again, we can clone. So something like a plant, for example, can make a new plant simply by cloning a piece of itself. But we can also undergo the ability to have sexual division, where we create a sperm and an egg, and we will reproduce by that. In that case, we'll be having eggs and sperm coming together to create live young. So we have different options. We can, and we'll talk about that again in a whole different chapter, but we can have sexual and asexual reproduction. However, organisms generally give rise to offspring of the same type. So for example, a dog is always going to give rise to a puppy. It's never going to give birth to an ostrich. And that's because DNA is faithfully passed on from parent to offspring. So even though the parents, like the mother and the father's DNA, may kind of combine into the offspring, it's never going to turn into an offspring of a totally different organism, such as an ostrich. And this is just depicting that all organisms, even the lower level organisms, are going to be reproducing. Here's a streptococcus bacteria. If you've ever had strep throat, you can thank this guy right here. Bacteria reproduce at a very quick rate, so approximately 2 hours, and they'll have a doubling time. So over 24 hours, you can have multiple sets of doubling time. So while you might not feel sick at the very beginning of an infection, it can flare up very quickly given the right conditions. As here is a dandelion. It's going to be producing seeds. They're going to get dispersed by the wind, land somewhere else, and create a new dandelion. So that's how one of the ways that plants are going to reproduce. And then, so that's by seed dispersal. Plants also reproduce by cloning, as I mentioned previously. And then here is an example of a mammal reproducing by creating haploid sperm and egg that then come together from parents, so from mother and father, to create a genetically unique individual here, this little panda baby. And this is the DNA that I was talking about. Don't worry, we'll have multiple chapters on DNA, so I won't go over it too extensively here, but it's a double helix. It's like a twisted ladder, you can see. And on the outside, you've got the little red balls. Those don't come on the inside. And on these inside, we have the little blue, right? So what are we seeing here? This is a sugar phosphate backbone. And these here are the base pairs that are going to be lining up with each other. We'll talk more about that later, but basically this is what is going to be passed on from parent to offspring and what can be subjected to evolutionary change under natural selection conditions. And as a whole, this is what I was just getting to, as a whole, organisms are able to evolve collectively, not the individual, but the population. And a population is defined of groups of the same organism living in the same area, and if they have selective pressure, whereby the external environment is causing a selection of certain traits or selection against certain traits, then we can end up with evolution or descent with modification from the pre-existing ancestral forms of life. And that's going to help explain all of the diversity of life on Earth. Don't worry, we're going to give an entire chapter, an entire lecture to evolution, so again, we're just glossing over it here. But just to kind of put your toe in the water, chimpanzees and humans are very, very different physically, or what we'll call phenotypically a little bit later on in the semester, and yet they share 95% of the same DNA. So they vary by less than 5%, and yet we have drastic phenotypic differences between chimpanzees and humans. In fact, there's even closer, less amount of differences amongst the human population that varies approximately less than 1%, accounting for all of the phenotypic differences of all of the humans that you see on Earth. Alright, so I just showed you a little bit of DNA, and we talked about evolution being defined as descent with modification. And the reason that that works is because natural selection works on the genetic variation in the population. So if a population is all the same, then if something comes through that is going to kill one, it's going to kill everyone, right? So we have to have genetic variation in the population, which means that when we have selective pressure from the environment, some of the individuals are going to survive. And they're going to have a higher rate of reproduction than those that, say, don't survive, which means that they're going to have a higher percentage inheritance of those variations of the parents that are able to adapt to or able to step up to the challenge of the selective pressure, causing an adaptation of the entire population. So natural selection is basically going to favor the individuals who are going to have enhanced reproduction due to whatever the genetic variations that they carry are, which means that they're going to have a better rate of survival and hence a better rate of reproduction. So again, we have to have variation within the population, and we have variations in the genetic material, and this happens because we have differences in the genes of the material. Genes are little segments of DNA that are the basic units of heredity. And as we copy them from parent to offspring, sometimes a mistake is made when we end up with a small change in the gene, and that's called, again, a mutation. So we can have mutations that arise during the copying of genetic material, but we can also have mutations that arise when DNA ends up damaged from something like cigarette smoke or UV rays from sunlight. So these are going to lead to things that might turn into cancer. For example, UV rays from sunlight have been associated with melanoma and cigarette smoke with carcinomas. But fortunately for the offspring, these are the kind of mutations that will occur in what we call somatic cells, so cells that are just your body cells and not cells that are going to be passed on, say the sperm and the egg, for example, to the offspring. So, for example, your grandfather smoking 3 packs of cigarettes a day is not going to give you lung cancer because it only affects his lungs and not his descendants' lungs. However, there are some mutations that get into the lineage which then do end up being passed down from mother to offspring. So some mutations are inherited, and those are going to be the mutations that arise in the sperm or the egg cells. And when we hear the word mutation, we think of it as being very negative. And oftentimes it is, but really most of the mutations are entirely harmless. Generally, we have a lot of play in the genetic code, and so oftentimes the mutation is going to end up with no effect at all. But most of the ones that do have an effect are going to have a negative effect and decrease in the organism's ability to function or death of the organism, or really just sterility would end up being the end of the line here. So an increase in the organism's ability to function would actually be the one slight benefit, is that every once in a while a mutation comes along that's going to increase the organism's ability to survive and reproduce. And when that happens, generally that mutation becomes prevalent in the population over just a few generations because it confers upon the organism that carries that mutation an increased ability to survive and reproduce. So natural selection is basically the interplay between the inherited traits that the organisms have and the environmental stress that is placed upon them in their particular environment. So whatever organisms are going to best meet their environmental conditions are going to leave the most offspring, meaning that those genes are going to get passed on at higher rates to the next population. But keep in mind that the environment might change over time or from different location to location. So the genes that might be favored in a particular habitat might actually not be favored in a different habitat, which is why organisms kind of tend to find their niche. And natural selection is going to help preserve the genes that help the organisms flourish inside the particular natural environment. All right, so how do scientists study life? We study life at multiple different levels. And the very smallest form of life is going to consist of just a single cell. So those are going to be unicellular organisms. Now, a multicellular organism, cells are going to combine into something called tissues, and then tissues combine further into organs, and organs into organ systems, and then multicellular organisms have multiple different organ systems. Think of your own body, for example. You have a digestive system, a reproductive system, right? A respiratory system, a circulatory system, a nervous system, a skeletal system, lots of different systems that are comprised of multiple different organs that all work together. Now, adaptations are defined as any structure that's going to help aid in the survival or reproduction. So anything that's going to make it that that organism has a better chance of reproduction than the other organisms in the population. And again, as I mentioned, adaptations that are good for one environment might be poor for another environment. So it's not just about having the physical characteristic, it's about having the physical characteristic when the natural environment is going to be in the favor of that characteristics. And if you can't rise to the challenge of the changing environment, then you can end up becoming extinct. And we have a lot of multiple extinctive events that have happened, but one of the most famous ones is going to be the extinction of the dinosaurs. Again, extinction is the complete elimination of a particular species. This here is either an Allosaurus or a T-Rex, something that once was a giant, terrible lizard and roamed the land and is no longer alive today. Now, some of the inherited mutations, again, they're going to help the individuals survive and reproduce, but since we have so many different habitats or environments in a different area, plus we have all of these adaptive processes, that's going to help us increase all of the biodiversity of the natural world. So that's going to be one of the major causes of species variety. And unfortunately, we as humans, since we accelerate the rate of destruction of our natural environment, we're also accelerating the rate of environmental change and therefore the rate of extinction of multiple different species. Now, evolution theory actually started with scholars Darwin and Wallace, and they devised a theory of evolution in the mid-1800s. And since then, we have a lot of different lines of evidence that have been found to support evolutionary theory. We have genetic evidence, we have biochemical evidence, we have taxidermy evidence, so lots of different types of evidence. And we'll talk about those when we get to that chapter. This diagram is talking about biological organization and complexity, and for the purposes of our class we will get a little bit into the species and population, but we're going to spend most of our time here talking about cells, tissues, and organ systems. So the first chapter that we're going to get into after this introduction is going to talk all about atoms. It's going to be our chemistry chapter and how these atoms come together to form molecules. Molecules are going to be a combination of multiple atoms. One of the most common molecules is going to be water, right? 3 atoms, 2 hydrogen and 1 oxygen. And then all of these different atoms come together, they form the molecules. These molecules come together to create this cell, which is going to be the smallest unit of life. Again, in order to be alive, you have to reproduce and have to meet all these parameters. And while some of the parameters can be argued for the molecular level, they can only actually all be met at the cellular level. We'll talk about the multiple different types of cells and how they have different functions, and then how they come together to make tissues, and how tissues come together to make organs, organs into organ systems into a multicellular organism like ourselves, like a human being. And then population would be multiple sets of the same species living in the area that are able to interbreed. And then we will talk a little bit past that, but we'll probably stop at the species level for the majority of our class. So we can study life at multiple different levels. So the next set of instruction that we'll talk about is going to be about atoms and elements, so we'll get an entire chapter on that, but just to kind of gloss over that, all of matter is going to be formed of what we call elements. And elements can be broken down into their very smallest units, which are atoms. So an atom is basically the smallest particle of that element that still retains the properties of that element. And atoms are reactive. They're constantly looking to create equal electron shells. And so they bounce off of each other until they combine with atoms that are like or meet their needs to form molecules. And these molecules come together. They're creating building blocks for cells, and cells, again, are the smallest unit of life that meets all of those parameters that we talked about to be able to be considered alive. Now, any of the organisms that are able to interact and interbreed, basically they're found in the same location and they're able to physically interbreed and create offspring, those are called a species. And any of the group of organisms of the same species in the same area, that's called a population. All the populations that interact make up a community, and then that's going to make up an ecosystem. So how else do we classify evolutionary relationships? We have three major domains. We have bacteria and eukarya is split into plants and animals. The fundamental differences among the cell types of organisms are reflected in the classification. So we are going to have, again, bacteria. We also have the domain archaea, which is that third domain. We're not actually going to talk on that for the purposes of this class. So we're going to focus on the bacteria domain and the eukaryotic domain. And the eukaryotes are going to be multicelled organisms like plants and animals, and also fungi. So bacteria and archaea are single cells, and eukaryotic organisms are multicelled. So basically we're splitting here into single-celled organisms, or unicellular organisms, and multicellular organisms. Now eukaryotic cells are going to be multicellular organisms, and they have at least one, but generally more, extremely complex cells. And the domain eukarya is split into three different kingdoms. We're generally going to be focusing on plants and animals, but just so you're aware, fungi also fall into the domain eukarya. So there's multiple different things that we're basically looking for in order to classify an organism into its domain and kingdom. One of the first things is going to be the cell type. Is it simple or complex, right? Is it unicellular or multicellular? And then also, how does it acquire its energy? Is it a heterotroph, meaning does it get its energy from the sun by photosynthesis? Is it an autotroph, which would be getting its energy from the sun by photosynthesis, or a heterotroph, which is going to be getting its energy from other organisms around that? I apologize for the mix-up on that definition. Again, we have here the domain bacteria and eukarya. Bacteria are going to be unicellular. They're prokaryotic versus eukaryotic. Prokaryotics are unicellular, and they can be either autotrophic or heterotrophic. So either they create their own energy or they absorb energy from other organisms around them. And then eukaryotes are split into plants and animals. Now plants, again, they're eukaryotic, multicellular. Both of these are going to be multicellular. Once you hit the eukaryotic level, we're talking multicellular. But plants, the major difference between plants and animals is that they're autotrophic or heterotrophic, meaning that they create their own energy from the sunlight or they ingest their food from their surroundings and other organisms. Now, we have multiple features of the cell that are going to be in common, even with bacteria and eukaryotes. So bacteria, again, they're prokaryotes. Eukaryotes are going to be things like plants and animals. But they're all going to have a plasma membrane, which is a very thin sheet of molecules surrounding the cell. The plasma membrane gets its own lecture, so we'll spend a good bit of time talking about what it's comprised of and what gets embedded in it. And then we also have organelles, which are inside the cell, which are the specialized mini organs, basically, the organs of the cell, that are regions or structures that carry out specific functions. And inside the cell, we also have DNA. Now, in a prokaryotic cell, it's not going to be contained in a nucleus, and in a eukaryotic cell, it is going to be contained in a nucleus. But DNA, in both cases, is going to be the hereditary material from which the parental cell is going to derive the offspring cell. Bacteria is going to be mainly prokaryotic cells. It's all prokaryotic cells. Eukaryotes are going to be all from the domain eukarya. And the reason that we're given the eukaryote versus prokaryote is whether or not we have a nucleus, right? So if we have a nucleus, then it's going to be a eukaryote. That's a true nucleus in Greek. A prokaryote means false nucleus or no nucleus. Eukaryotic cells are much larger than prokaryotic cells. They're generally going to be found in multicellular organisms. They also have a variety of organelles and specifically the nucleus. So the prokaryotic cells are anucleated or do not have a nucleus. Most eukaryotic cells do have a nucleus. However, in the human body, the blood cell is going to be an exception to this rule in that it is anucleated or does not have a nucleus. We'll talk about that a little bit later, but generally, cells that are eukaryotic are going to have a nucleus. And they're all going to be found in the domain eukarya, and they're much more complex than the prokaryotic cells. Prokaryotic cells are before the nucleus or false nucleus. They are much smaller, approximately 2 micrometers in diameter. In contrast to this, the very smallest of the eukaryotic cells is about 7 micrometers in diameter. So they're much, much smaller even than the red blood cells. They lack organelles, so they don't have any membrane-enclosed organelles. They don't have a nucleus, and they are generally going to be bacteria. Again, they're much, much smaller, much more simple than eukaryotic cells, and they probably arose first in the evolutionary lineage. So unicellular organisms are found in bacteria, and multicellular organisms are plants and animals and also fungi. All right, so I feel like I've hammered this home, guys, so this might be on the exam. Eukaryotes are going to acquire their energy in 2 different ways. One, they get it from the sun. That's autotroph, a self-feeding. That's all photosynthetic organisms, basically anything that's going to take energy from the sun and store it, generally in sugar, so in glucose molecules, but also in fats. This is going to include almost all plants, some bacteria, and some protists. And then heterotrophs mean other feeding, or organisms that get their energy from other sources by ingesting molecules from the bodies of other organisms. This is going to include archaea, a bacteria, and then also fungi and animals. And all organisms are going to acquire food, but the size of that food can be variable. Sometimes there are much, much smaller individual food molecules, and then we as humans or larger organisms will eat chunks or sometimes even entire organisms in one bite. So how do we name organisms? We have a hierarchy of different categories, and this includes something called genus and species. And genus is going to include multiple different species that have very similar characteristics. And species include organisms that can interbreed. This is very important. As soon as a species can no longer interbreed, then they're considered 2 separate species. So in order to be considered the same species, you must be able to interbreed successfully, right? That means that you must have offspring that are also able to reproduce and are nearly identical or identical in many different ways. So we use a binomial system. That means 2 names, right? And the genus name is going to be capitalized, and it's going to come first. And for us as humans, we're Homo sapiens, right? Both the genus and the species name are italicized, but the species name is not capitalized. And you may see Homo for a whole different lineage all the way back, Homo erectus, for example. So you'll see Homo as the entire category of a lineage of species, which for us would be Homo sapiens, but we're descended from other Homo organisms. All right, so what is science? Science is an inquiry. We're basically looking at a world around us, making an observation, and then making a statement that we believe to be true. But in order to actually assert that something is true, we have to run an experiment. And we run the experiment, then get the results of the experiment, and then can demonstrate that something is true or is false. And we're basically, through this systematic inquiry, looking into the origin, structure, and behavior of our entire environment, be it the living and the non-living part of our environment. And science is based on the principle that all events have natural causes, right? And we have to throw in a corollary here that we don't have any supernatural forces, and that everything that we gather has not been deliberately distorted, that the information that we have can actually be traced back to natural causes that we can comprehend, or natural causality. And we'll talk extensively about the scientific method when we get to our laboratory section, but there are 6 main elements of the scientific method. The first is observation. Basically, you look around and you see something and you question, why is that the way that it is? So you observe something that you see to be a fact, and you question why that fact is true. Then you make a hypothesis that you believe this to be true because. Okay, so for example, I observe that the plants around me are green. So the question in my head becomes, why are the plants around me green? And then I make a hypothesis. I'm going to make a ridiculous one. The plants outside are green because little tiny leprechauns come around and paint them green at night. Well, if that's true, so now I'm going to make a prediction. If that's true, then if I set up video cameras, that I should be able to catch video footage of the little leprechauns running around and painting my leaves green at night. Right? So then I'm going to run that experiment. So I do so, and at the end of the day, at the end of the next day, I check the video footage, and it turns out that I don't have any video footage of little leprechauns. So then I get a conclusion, and the conclusion in this case does not support the hypothesis. The conclusion is, well, unfortunately, the leaves are green, but they are not green because little leprechauns paint them green. That brings us back to our hypothesis. We have to come up with another reason why we think they're green, and then we can run with that prediction and then run that experiment, right? So we have multiple different levels, but they're not a linear process. It's not 1, 2, 3, 4, 5, 6, right? It's 1, 2, 3, bounce around between 3 and 4, run from 4 to 5, get to 6, and then maybe bounce back to 3 again, right? So these are interrelated elements until you finally get to a conclusion that you feel that you can stand upon. It's a very rigorous method for making observations, again through scientific inquiry, where we're going to take an observation of a specific phenomena and ask a question. Again, why are the leaves green? Then we're going to formulate a hypothesis based on the fact that the leaves are green, and we think that perhaps it is XYZ, and we're going to offer that up as an explanation for why this is happening. In order for us to be able to say that that's true, we have to predict the the expected outcome if the hypothesis is correct, and then run the experiment and see if we get the predicted outcome. And we do this by an experimental design whereby we have very specifically controlled manipulations. We only change one variable at a time, very carefully designed experiments where we collect data by running specific parameters and observing what occurs and seeing if we can get that back to the prediction of the expected outcome. And if we do, then it's going to support the hypothesis. And if we don't, because we weren't able to get the footage of the leprechauns, then it refutes the hypothesis, which is going to allow us to develop a conclusion, right? The conclusion is the data support the hypothesis, the data does not support the hypothesis. And we do this again by having controlled experimental design. So we basically are going to have one control and one variable. We only change one variable at a time. So we have a control situation where we hold as many variables as possible constant, and then we have an experimental situation where we change one variable to test the hypothesis to be able to determine that the variable is the cause of whatever it is that we observed. And last but not least, science is only important if we're able to communicate it to the scientific community. And so science has a very rigorous testing method whereby it presents itself out to the scientific community, and then the community is allowed to shoot as many holes in it as possible until it finally weathers scientific review. But if you know something and you die holding on to that information is completely useless to society, again, unless you're able to communicate it. All right, so this is an experimental design that's a very simple experimental design, but it follows all of the 6 steps. So the first, we're talking about flies here. So we have a jar, then we have meat in the jar, and we have flies. The question that the person is asking is, where do maggots on meat come from? And so he notices, the observation is that we see that flies swarm around meat, and also that maggots appear on the meat. Where do the maggots come from? The hypothesis that flies produce the maggots. And now we all know this to be true, but at the time, it was a big question, right? So the question was, if the flies produce the maggots, right, then if we keep the flies away from the meat, will it prevent the appearance of the maggots, right? And it ought to if the hypothesis is correct. So how do we do this? What's our experimental design? We have two identical pieces of meat and 2 identical jars. The only difference is that we're going to put gauze on top of this jar and no gauze on top of that jar. So the experimental variable here is the gauze, which is going to be preventing the entry of the flies in this one and not preventing it in that one. So this jar is uncovered. This one is covered. Over multiple days, we look at our results, and lo and behold, the maggots are going to appear where the flies were swarming and not appear where the flies were kept off of the meat. So the conclusion is that we're supporting the hypothesis. Flies are the source of the maggots, and that maggots do not spontaneously generate, which was the alternative option for the hypothesis, is that maggots actually come out of the meat. Here's another example of an experimental design. If we observe that male widow birds have very long tails, then the question becomes, why do males have long tails but not females? And the hypothesis is that it's sexual selection. Basically, the males have long tails because females prefer to mate with long-tailed males. That leads us to the prediction. Basically, if females prefer the long-tailed males, then males with artificially lengthened tails should attract more mates. And a sub-hypothesis that's not written here, sub-prediction here would be that the males that have shorter tails will attract less mates. And so what do? They divide the birds into 4 groups. One group has no manipulation to the tail. The second one has manipulation to the tail in the fact of cutting and re-gluing, just to see if the act of cutting and re-gluing changes it. Maybe, for example, the females hate the smell of the glue, and none of them that are manipulated are going to have any mating effects. And so, therefore, they want to make sure that the actual manipulation is not going to affect the scientific results. So these guys, they have no change to the tail. These, we have the tail is cut off and glued back in place, so we have had the surgery basically, but we're going to have the exact same phenotype. And then these guys here have had half of the tail taken off to half the length, and this one is going to have the feathers from this one added to double the tail length. All right, so what happens at the end of the results? We have about 1 nest per male for the original, and also for the one that's been manipulated but hasn't had any length or shortening changed. And then we have less than half a nest per male for the ones that have had the short tails, and about 2 nests per male for the ones that have the large tails, or the long tails. And that indicates that the hypothesis is supported, right? That female widow birds do prefer to mate with long-tailed males and are less likely to mate with short-tailed males. Okay, so that's kind of the overview of scientific design. Now, when someone says scientific theory, I know we tend to scoff at it, like the theory of evolution, but please keep in mind that we still have the theory of relativity, right, and theory of gravity, and scientific theory is actually a much stronger term than the word theory that we use in everyday life. In science, a theory is defined as a general and reliable explanation of natural phenomena that has been developed through extensive and reproducible observations and experiments. Over and over and over, extensive and reproducible. So it's not something that someone just came up with on a whim. It's something that someone thought of, ran experiments, presented it to the community, allowed the community to try to shoot it down, and eventually ended up to the point where it was unable to have any more holes poked in it, and therefore was solid and sound. So that is actually closer to a natural law than a theory, the way that we use theory in colloquial language. It's a basic principle derived from the study of nature that has never been disproven by scientific inquiry. So for example, the cell theory has never been disproven. The cell theory is that all living organisms are composed of cells. So no matter what organism it is, if it's alive, it's composed of cells. Right? Another theory is a theory of evolution. And these are very fundamental to the study of biology. So when we use the word theory, we don't use it lightly. We also have a principle of natural causality, which is basically stating that all events can be traced back to natural causes, and that the same natural laws are going to apply every time and everywhere. And scientific inquiry also is based on an assumption that everyone is going to perceive a natural event in a similar ways. And this is why we have to take our science and put it out to the, maybe not the general public, but to the scientific public, to allow people to poke holes in our theories until we finally get to the point where we're standing on the truth. Because the point is to throw as many ideas out there and then whittle away what's false until everything that we're standing on is true. And sometimes we have to revise our thinking very dramatically when new scientific evidence comes to light. I don't know if any of you have heard of the term prion, but prion is basically an infectious protein. And prior to the discovery of prions, everyone thought that proteins had a function that was determined based on their structure. And that is true, but it also turns out that they can be infectious if they're able to change the structure of the proteins next to them into the form of a protein that has the primary function of changing the protein of the one next to it. And in this way, protein A changes protein B, B changes C, etc., until all of the proteins of this category have been changed into infectious agents, basically. But it was always thought, and with everything except this exception, it's true that infectious agents have to be RNA or DNA based. So before we knew anything about prions, all infectious diseases contained DNA or RNA. In the 1982, Stanley Prusiner demonstrated that we actually have a protein that's able to be infectious. It's called a prion. And one of the examples of this is mad cow disease in humans. There's another there's several different types of these proteinaceous infections. But my point is that scientists were completely unwilling to accept his information until they actually had the data rubbed raw in their face. In order to be able to allow this understanding and expanding of study of prions, we had to make it to the point where the scientific community was able to and willing to listen to the data. So we have multiple different ways of scientific reasoning. We have inductive reasoning and deductive reasoning. And inductive reasoning is basically going to be helpful in the development of scientific theories by creating a generalization from multiple different observations that support whatever that generalization is, and none that contradict it. So for example, if you pick up a pencil and you let go of it, it will fall to the ground. And it will happen 100 times out of 100, 1,000 times out of 1,000, 100,000 times out of 100,000 times. So you can make a generalization that every time that you drop your pencil, it's going to fall towards the ground, right? And that's going to lend to the theory of gravity, for example. And the theory of gravity is that Earth is going to have a gravitational force or a pull on objects that are going to fall downward towards Earth, and absolutely no observations of objects that are falling upwards away from Earth. Now, deductive reasoning is a little bit more expansive. It's going to be taking a well-supported generalization and expanding it to new information. So for example, the cell theory has never been disproven. And so if we find a newly discovered organism, an extension of cell theory would be that that new organism would be expected to be composed of cells as well. And that's called deductive reasoning. So it takes a little bit more of a leap. And we call the principles of science theories because theories are able to be disproved or falsified. But when we get to the point that they are theories, they haven't yet been disproven. It's just that we are able to disprove them perhaps if new information comes to light. But when we falsify theories in scientific theory, it's very, very different between scientific theory and faith-based belief, because we're able to use scientific theory, we're able to use facts and something that we can stand on. We don't have to make any sort of leaps of faith. There is no belief. It's a solid information. So for example, the faith-based belief that each creature on Earth was separately created, we actually can't subject faith-based beliefs to scientific inquiry because it's a belief that's rooted in faith and can't actually be tested in any way. Now, one of the cool things about science is that science is a uniquely human endeavor, and therefore human personality traits are actually part of real science, and science ends up being driven by all sorts of things, sometimes good things, sometimes bad things, right? Driven by pride, ambition, fear, etc. And the scientists both make mistakes and get lucky, right? Accidents and lucky guesses, etc., can sometimes contribute strongly to scientific advances. One of the classic examples is the invention of the microwave. The guy who was using the microwave was actually trying to send radio signals. It was just trying to send it on a very far out frequency, and instead he ended up melting a bunch of chocolate and was, although annoyed by the mess, was eureka, onto something, and ended up inventing one of the ways that we all heat food in our house today. So that was just an accident. And what's interesting about that is that you have to be prepared to completely shift your mindset if you're given data that is completely off the topic of what you were actually going after. An example of this, for example, is Alexander Fleming. Now, Alexander Fleming was attempting to work with bacteria for bacterial studies, and one of his bacteria cultures became contaminated with mold. Had he just thrown that bacteria culture away, we would never have had penicillin. Well, perhaps not never, but not at that time, because what he noticed is that the mold inhibited bacterial growth. And he said, wait a second, that means that the mold might produce an antibacterial substance, and lo and behold, he found penicillin from the Penicillium culture, which led to the first antibiotic, or penicillin, right? And this is just a depiction of that. So here's the bacteria on the outside are red or healthy, unhealthy or white, and then this in the middle, there's no bacteria growing at all. It's the mold that's actually growing the penicillium, and this means that it's competing for the same nutrient source, and so it's killing off, basically, it's releasing an antibacterial compound that's killing off anything that's competing for that resource. Now, had he just thrown that away, he would never have discovered it. Had the person just tossed out the chocolate that was melted and been annoyed with it instead of being able to use that to develop the microwave, then we would have lost that information. So not only do you have to be prepared to study what you're looking at, but you also have to have an open mind to everything around you, because chance really favors the prepared mind. And in that way, it's really interesting to just kind of pay attention to the fact that science is a very human activity. Even though we're looking at the study of life, we're actually looking at it through the lenses of our own human endeavors. Anyway, that concludes our lecture on life on Earth, so I appreciate you showing up to class today. Happy studying, everyone, and have a fantastic day.